Welcome to the latest episode of Star Cells and God. My name is Fuz Rana. I'm a biochemist and a Christian apologist, and I'm joined in studio today by Dr. Robert DiSilvestro, who is a biochemist and a professor emeritus of nutrition at The Ohio State University. And we're going to talk about the origin of life question. Can protein folding give us insight into how life originated through chemical evolution? Bob, welcome so much to Star Cells and God. Glad to have you.
Good to be here. I have a very diverse background. As you mentioned, I consider myself a biochemist. I have an undergrad and a PhD in biochemistry. I was a professor for many years in nutrition, and my research ran the gamut from what I call real biochemistry all the way up to clinical nutrition research and nutraceutical research. So I've done a lot of different things, but I've been very interested in this origin of life issue.
Yeah, well, that's something I've been very interested in myself because there are some really significant implications of the origin of life question to the scientific case for the Christian faith. But first of all, just to clarify for our viewers and our listeners, what is meant specifically by the origin of life question or the origin of life problem?
Well, today, life always comes from other life. Right. So it raises the question, where did the first life form come from? Right. So natural, biological, biochemical researchers will say if there's a natural explanation, then life had to come from non-life. The other alternative would be a supernatural being we call God started life. Yeah. So if it's not possible for life to arise from non-life purely by natural explanations, then that's a strong argument for the existence of God.
Yeah, yes. And so, this is not a trivial issue when it comes to building a scientific case for the Christian faith, or at least for the role of an intelligent agent in bringing life into existence. Because the mainstream scientific view is that life emerged through some kind of chemical evolutionary process. But I also think it's safe to say, too, that origin of life researchers will be pretty quick to admit, both privately and publicly, that they really don't know how life could have emerged through chemical evolution.
Yes, that's true for the people that are actually doing the work, but sometimes I look at internet chats and I see people on there saying, well, the origin of life has all been figured out except for a few minor details, and we don't need God. And I look at this and I'm thinking, where do you get that from? Yeah. Look at what the actual people that are doing the actual research are saying. And they're saying there's just some tremendously difficult challenges in trying to explain life's beginnings from just purely natural processes.
Well, I've had the chance to hang out and interact with some of the origin of life researchers who are at the cutting edge. And they're a fun group of scientists to hang out with because they relish in the fact that the problem is extremely hard. And in fact, I almost get the impression that if they somehow figured out how life originated through chemical evolution, they would be deeply disappointed because there's no longer this fun problem to solve. But they also are brutally honest in terms of really where the discipline is at in terms of understanding the origin of life question. I don't know if that's your experience as well.
I think they're way more honest than, say, evolution of species, where if you challenge that, a lot of people get very emotional and say, no, we don't need God to have made the different species. That happens through evolution, and they won't even talk to you about the problems. Whereas I notice in the origin of life literature, they are a little more, as you say, direct and honest. In fact, the paper I'm going to talk about today, the very first line says the following: no one knows how life originated. Yeah. That could be more direct.
That's right. Well, and let's get to this paper because, origin of life researchers, even though they, again, will say, look, we have no idea how life originates. We might have clues here and there or small isolated pieces of the puzzle, but we really don't understand how life originates. But it doesn't mean that they just abandon ship. They are continuing to present ideas, some of them very clever ideas, on how they think life could originate. And the paper that you want to talk about essentially is in that category, I would say. So tell us a little bit about who the author is, the title of the paper, where it was published, and then maybe a little bit about the work and the nature of their approach.
Yeah, the paper is called Origins of Life, the Protein Folding Problem All Over Again. It was written by Charles Coker and Ken Dill. It was published just this past year in the Proceedings of the National Academy of Sciences, very prestigious journal.
Yeah. And Ken Dill is a very well-known protein chemist.
Yes, and I feel a little humble in trying to critique his work in some ways. But this paper only addresses one of many problems in explaining the origin of life. And they're upfront about that, the two authors, and saying we're just looking at one particular issue, not everything you could possibly bring up. And this one particular issue involves proteins. Now, when I say proteins, in some people's minds, they may think Premier Protein you buy at Sam's Club, or maybe the contractile proteins in muscle, and that is protein. However, some people may not realize that all forms of life have hundreds and hundreds of proteins, and they do all kinds of things, and a lot of them are enzymes. Enzymes are proteins that promote certain chemical reactions, cause them to proceed at a reasonable rate in life, and anything that happens in life requires enzymes.
Right.
So that's what I'm talking about when I say proteins. So even the simplest form of life that we know of now has close to 400 functional proteins. So one of the questions is how do we get to that point where you have these functional proteins? So proteins, as you know, Fuz, are made of building blocks we call amino acids. Mm-hmm. And now most proteins are commonly made of 20 amino acids. And they have to be arranged in a certain order to give it a function, or to give it any function, really. And most of the random sequences don't give a function.
Right.
Now, you can make a few little substitutions. On my slide, I show a chain of amino acids here, a short chain. And in some places, there's alternatives underneath. For a few amino acids in a protein, you can have a little variation. And that's why sometimes for, say, a rat protein versus that same protein in humans, there may be small differences. But most of it has to be the same for the protein to function. So you can think of amino acids a little bit like letters.
Yeah.
They're arranged in a certain order, and that gives us words and sentences and books.
Right.
But if you just start arranging letters at random, you get gobbledygook. And the same thing is true with proteins and amino acids. They have to be in certain orders to give any type of a function, and most of them don't. So the question this paper is addressing is, out of all the, literally billions of possible arrangements you could have of amino acids, how did the first form of life arrive at a point where they just settled on, these forms of life just settled on a few hundred sequences and got those instead of all the ones that didn't work? So that's the question they're trying to answer.
Right. And just to build off of your point, you know, a typical protein, I think in your diagram you're showing a chain of about 30 some amino acids. In a typical protein, you're dealing with probably about 300 amino acids, ballpark. That would be an average protein, wouldn't you say?
There are different sizes, and for instance, not in grad school, but as a postdoc, I worked with a mineral binding protein called metallothionein. It's really short, but most of the proteins are much longer, like you're saying.
Yeah. And it's not just the sequence, though, too, because the backbone adopts specific structures, you know, alpha helices, beta pleated sheets. People may have heard those terms. And those in turn fold to create a three-dimensional structure that's really vital to the protein's functional attributes.
Yes, protein folding is pretty much the first thing you have to have for function for most proteins. So that's a minimal requirement. Right. So this paper talks a lot about protein folding. So here's their solution to this problem of, gosh, there are billions of sequences. How do we arrive at one that or one series that would go into the earliest forms of life. And they draw an analogy to evolution. In evolution, there's the idea of natural selection, that over time, different genes get changed where they'll have a small mutation. Many of these are very deleterious, but sometimes they can actually improve the protein that the gene is coding for, and that this can make certain improvements, and that can eventually lead to a certain species taking on more of the beneficial improvement of this protein. So, for example, if you have moths in a certain part of the world, there's people that question this one, but it's one that comes up a lot in evolution discussions, that when there was pollution, the moths that had proteins that gave it a white color could hide on the trees from their predators better because the trees were kind of white. Well, when the pollution was cleaned up, that population declined, and the ones that had a color that blended in with the more natural foliage colors, they predominated. And that does work to some extent. I debate the idea that can go on and create whole new species, but you do have a little bit of natural selection in nature where a given trait will predominate when that's favorable, and that trait will be passed on through generations because that trait gives a certain reproductive advantage. But before life began, you don't have that.
Right.
So these two authors basically say, okay, is there something like that could have happened? And they said yes, and it's basically about protein folding. So this is right out of their paper pretty much. I put a few little commentary text spots in there, but basically what this slide is showing is that in pre-life there could be some mystery green box here in this figure that made the proteins. They kind of downplay how difficult that is.
Right.
Today, when proteins are made, they're made in life, or at least borrowing the machinery from life to make those proteins. Trying to make proteins purely by chemical means is slow, and it's hard, and so far most of the methods...
And it requires virtually anhydrous conditions where there's no water around whatsoever.
And they use conditions you couldn't have had in early life. Like they'll put a chemical in, and then when that does its thing, they pull that out and put a different one in. So some people would say that, well, that's a deal breaker right there. You can't even make these chains of amino acids. But they have this, as I said, this kind of a green box they call the founding rock, whatever that is, to somehow put the amino acids together. And they also assume the amino acids are there and you have the right ones in the right geometry, and that's a whole can of worms in itself. But as they said, they're only trying to tackle one particular problem at a time. So they're saying that these different chains would form. Some of them won't fold, and that's what's up on the top of the figure. Others will fold and form small chains of amino acids with folds, and that the process keep going on and build on the ones that are folding until you get bigger proteins that have some practical functions. So how do you get the advantage of preserving the ones that are folding and the ones that aren't? Well, it all has to do with breakdown, also known as degradation. The idea here is that the ones that don't fold will break down very quickly, and the ones that do fold will stay around longer. And they give some mathematical models where they show these are predictions that can be made about the speed of the breakdown. And I will say the paper was very well written. Even if you didn't understand some of their mathematical calculations, they were very good at explaining the big picture idea. And their big picture idea was that the unfolded ones would break down and the folded ones would be preserved, and eventually you would end up with some very usable functional proteins for the first form of life. That's the basic idea of the paper, and kind of interesting, and I thought their model was very elegant, but I have a lot of problems with their conclusions.
Yeah, well, okay, so, I mean, I can sit here thinking myself of a lot of problems that we could raise with, with their conclusion. But it does seem to me to give these two researchers the benefit of the doubt that they have identified at least a mechanism to create persistence in certain proteins and certain amino acid sequences?
Well, the first issue is their math. And I got to admit, I couldn't follow every little bit of their calculations, but it somewhat reminds me of when I do statistical calculations before I do a clinical study. As I said, I have a very diverse background. And I sometimes have done clinical intervention studies. And we have to do a calculation ahead of time to think, how many people do we need in the study? And you can do these calculations and you get a very precise number that says, okay, if you use this many people, you have this probability of getting meaningful results. And the calculations are very, very precise. But the numbers I put in there are totally subjective. And this reminds me a little bit about that, gee, they get these calculations that predict these degradation rates, but they don't actually know what the degradation rates were when life first started. You've worked with enzymes, I've worked with them, and to preserve them when you pull them out of life and put them in a test tube, you have to do things like sometimes you can freeze dry them and the powder will last for a while. Other times you do have them in a solution, but you put it on ice and you add different chemicals to keep it stabilized, and you use a certain buffer. Right. Well, in pre-life, you don't have that. Who knows what was in those solutions they were in, and it could have promoted breakdown extremely fast. But even today, under ideal conditions, we have enzymes that I used them one day and threw them out the next day because they went bad. Yeah. So as I show my next slide, we got what I call the A, the non-folded, and the B, the folded. Right. And for this model to work, two things have to happen. First of all, the breakdown of A has to be much, much, much greater than B. Yeah. So it has to be really a big difference. Right. Secondly, B has to be very long, even if there's a big difference. Let me illustrate it this way. Let's say that A breaks down, half of it breaks down every minute. Well, let's say for B, half of it breaks down every 5 minutes. That's a five-fold difference, but it's still not very long. Right. So you have to have a situation where B breaks down way, way, way slower than A, and that it lasts a long time. And I would say it'd be very hard for most proteins to last very long because, as I just said, even today, where we're extremely careful, we could have enzymes lose their activity in literally minutes if we don't do certain steps. So I think that's my first problem with this, is that their numbers are very nice that they get, but they're feeding in completely theoretical values to start the calculation, and we have no way of knowing that the breakdown rates would really be what the numbers predict, and I think it's actually worse than that. I think it's very likely that all the proteins broke down fairly quickly, or at least most of them.
Yeah. Okay, so then what else do you see as issues with the study?
Well, another thing has to do with specific functions. Just folding isn't enough. Yeah. And you need very specific functions. Now, I used to sometimes in my talks put up these enormous numbers of saying that certain complicated proteins, the odds of getting the amino acid arrangements right were like 1 over 10 to the 64th power. And everybody go, whoa, everybody's really impressed with that. Well, one of the arguments I heard back to that was, well, in the beginning of life, you didn't necessarily have to have proteins that were that specific and that complicated. You could have had some smaller ones. Right. But actually, that's partly true. But even using that logic, you still have to have an awful lot of specificity. And I recently wrote about this in an apologetics site I started. And I went back to an experiment that I did read very carefully. And it was an experiment where a group of researchers made proteins. Now, they didn't make them just purely chemically. They borrowed biological machinery to make the proteins, and they could generate a whole large number very quickly. And they said, okay, how many of these randomly generated sequences would have any function? So they set the function bar very low. I wouldn't even consider it function. What they were saying was binding ATP, and that's not really a function in itself, but that was what they did. And they still came up with only about 1 out of 10 to the 11th power sequences even would do that.
Yeah.
And they concluded the paper by saying, and then there was a, not only in the paper, but also in an interview that was published, they basically said, getting order from randomness is really hard.
Yeah.
A contrast to this PNAS paper that basically said, well, with this folding, you can get order from randomness really easily. I don't think so. And this was saying that for real highly specific function, you need a lot more than folding. And you need the right functions, and you need them all there at the same time in concentrations that are high enough to do something.
Yeah. Well, I think you're on to a really significant point here because, have spent quite a bit of time looking at studies with regard to the requirements for minimal life. And as you said, minimal life could potentially have as few as 400 different proteins, but those proteins not only have to have specific functions, and most people think minimal life would have had to have the ability to replicate DNA, to produce proteins, to harvest energy from the environment, and to build some kind of cell wall or cell membrane. But that means you have to have specific functions, and those 400 proteins not only have to have specific functions, but they have to be working collaboratively in order to achieve even the basic requirements for life. So this is not a trivial problem whatsoever.
Yeah, there was a Harvard website that I looked at just the other day and talked about origin of life, and the site made the comment that in the cell there are chemical reactions, but those chemical reactions aren't life. Somehow you put all this together and something happens that's different than just the sum of individual chemical reactions, and that's still very mysterious.
Yes, it is. Well, and something else too, and not to preempt your thunder here, but let's just say for the sake of argument they're correct, right? That you wind up by getting proteins to fold, that they somehow have stability. Wouldn't it be a remarkable set of coincidences for those folds to actually be exactly the folds that you need for life to be possible? So if you grant their argument, you almost are forced into a type of design argument, are you not? Even if you see it from an evolutionary perspective.
Yes, it would be a little bit like the so-called anthropic principle, where the conditions of astronomy and the physics constants all work out in a certain way that we can have life. It's a lot like that, where it would be saying that even if this could happen, which I still dispute, it would say, boy, what a coincidence.
Yeah.
But it's really, you really can't say it would be coincidence, because it would be so ridiculously unlikely that we would be this lucky, not just in one respect, but in the respect of having all these proteins show up that way at the same time.
Yes.
So that leads to a third problem I have. Now, they make the assumption in this paper that once you preserve certain proteins and they fold, that you can keep building on them. And one of my problems with a lot of origin of life discussions and evolution discussions is there seems to be this assumption that if you keep building, it'll just keep getting better. And there isn't built into the thinking the fact that as you're building, something can happen that can get you all the way right back to ground zero. The whole thing could fall apart. I put this little very scientific-looking illustration up here, but I think most of us, when we were younger, stacked things.
Right.
I used to stack cans of vegetables, or you do blocks, and you know that the higher you get, eventually you're going to put one more on, and what happens? Boom. It all falls down. And so they don't really take into account that the odds are very strong that before you get very far, something's going to happen that's going to mess the whole thing up. So yes, the proteins might fold. I wouldn't even call them proteins. I call them polypeptides, which are shorter chains than a longer protein. As they get bigger, the chances are extremely high that you'll start getting a sequence of amino acids that wouldn't fold, and that'll start breaking that down, and you go back to step one. This is different than natural selection. Natural selection doesn't preserve proteins. It preserves the genes that make the proteins.
Right.
So if you have a gene mutation that makes a protein that now works better for the current environment, you don't preserve that protein. You preserve the gene,
Right.
and the cells reproduce, and the gene is preserved, not the protein. So in the primitive world before life existed, once the protein breaks down, you're back to zero.
Yeah.
You know, you can't just say, well, the environment remembers we had a protein here that worked well and it folded.
Right. Yeah.
You're back to zero and having to start the whole random process all over again. So I really see a problem with preservation here, because I think the odds would be incredibly high that you would, after a few steps in trying to make an elongation of these short chains, you would eventually lose the folding, and it would break down faster, and you go back to the start, and there's no memory built in that could reproduce that the way you would have in a living system where you can preserve the genes when the cells reproduce.
Well, you know, in a sense, you're never going to be able to have true reproduction, which is the point that you're making so ably. Because let's just say, hypothetically, you somehow encapsulate this collection of proteins in a membrane-bound system, right? And you might have even multiple copies of the same proteins. Well, you can, by accumulating, additional membrane-forming materials into the membrane, eventually it'll fissure into two daughter membranes. And you might statistically get two daughter protocells that have roughly the proteins that they need, but there's no way, eventually that process is going to fizzle out, even if you have infinite preservation of those proteins. So there's no way to truly reproduce the system, is to your point, without having some kind of genetic material that's encoding those proteins.
And also, proteins can get in each other's way. If you had a non-functional protein, it could actually get in the way. That's something that I do in some of my research, where I've done a little bit of drug design, and there's always this concern: if we get something that looks like this but doesn't work like this, it's going to get in the way of what we're trying to accomplish, and that's often not taken into account. And a number of years ago, there was someone from, I believe, Michigan State that gave a seminar at a university that was not mine, but I went to the seminar, and they were using computer simulations. It was for evolution, but I think the same principle applies to some origin of life issues, where he was building proteins on the computer. And I gave a little rebuttal seminar for a Christian group a couple weeks later, and the people from the biology department all showed up to hear what I was having to say, at least the professors did. And I said, the problem with these computer models is they're rigged to keep going forward. There's nothing in the model to destroy everything and go back to square one. And that's what happens in real life, or well, no, I can't say real life because I'm talking about before life existed, but in the real world, let's put it that way, that these models often don't build in for the possibility that everything's going to go back to dust, essentially.
Yeah, that's a really good point. And this whole idea of, as you're pointing out, if you continue to build upon a folded protein, it could start to unfold. And, if you're in an aqueous system, those residues that are internal are going to be hydrophobic. And once they get exposed through unfolding, then they start latching on to other functional proteins and literally just create havoc, right? It just creates protein clumps in the cell, right? I mean, that's the problem with when proteins denature in a living cell. It's not a good thing.
Yep, that's correct. Well, I know what a lot of people will think when they hear this. They'll say, well, you want to bring God into this whole thing, and isn't this just another God of the gaps?
Yeah.
So if you're not familiar with that expression, let me explain it. Way back in history, when people couldn't explain why something happened, they would just say, well, God did it, so why did it rain today? Well, God made it rain. Now we have meteorologists telling us, well, it rained because this pressure front hit this one, and we have natural explanations, and sometimes when they predict the weather a week from now, they're even right occasionally. And so we have known reasons why it rains. So God of the gaps was at one time the explanation. God just, opened the spigots of the clouds and it rained. So some people are going to say, isn't this just another God of the gaps argument? But it actually isn't, because it's not based on what we don't know. It's based on what we do know.
Yeah.
We know a lot of things. Do we know everything? Of course not. But we know a lot of things about how molecules preserve, how molecules in biology are made. And when we look at what we do know, this just seems completely implausible, this being the idea of an origin of life from purely naturalistic mechanisms. Comes out, in my opinion, very implausible. And we only covered one little part of the problem here. We didn't, for example, talk about chirality, where amino acids and many other molecules in the body have to be either left-handed or right-handed. And when they're made by enzymes, you get the right version. When they're made by pure chemical reactions without enzymes, you get a mixture of lefty and righty.
Right.
And that itself is a very daunting problem.
Well, and let's just say hypothetically you had, what's called a racemic mixture, a 50-50 mixture of left-handed and right-handed amino acids. You're never going to get proper protein folding, because everything has to be either left-handed or right-handed for the functional groups of the amino acids to be in the right orientation and space to stabilize the protein folds. So you're not going to get folded proteins with racemic mixtures.
And I also thought it was interesting that in this paper, they said, well, eventually you don't need the founding rock anymore. Eventually, you will make proteins that can make other proteins. I thought that was an interesting comment, because in real life today, single proteins or even a family of enzymes can't make other proteins.
Yeah.
In real life now, when proteins are made, yes, there are enzymes involved, but there's also messenger RNA and ribosomes.
Yeah.
And you don't have all that. I didn't talk about this in my slide, but just having proteins isn't enough. There has to be something for the proteins to work on, and there has to be these cooperative molecules for the proteins to make other proteins.
Yeah. Well, in fact, proteins are made at ribosomes, as you well know, and the functional part of the ribosome, at least with regard to forming the amide linkage or the peptide bond, is actually an RNA molecule. So ribosomes are ribozymes, which are RNA enzymes. And in fact, that's one of the reasons why many original life researchers appeal to the RNA world, where the very first biomolecules were RNA-based, for some of the issues that you're raising here with this work.
But the RNA world has its own set of problems, and in fact, the authors of this paper pretty much dismissed that right away and say, no, we couldn't have started with RNA. We have to start with proteins.
Yeah.
So that was interesting. But again, I just want to go back to this whole God of the gaps thing. That this is not just inserting God into a complete black box. This is saying we know a lot of things, and based on what we know, natural origins of life isn't plausible. Yeah. Now, that doesn't mean that researchers can't look, and I know someone will say, well, what if we discover something next week that really helps us? I said, what if you discover something next week that makes the situation worse? It's a faith statement, a blind faith statement to say you're going to discover something brand new. And there's a point at which we're not trying to prove God started life here. We're trying to say that the current evidence strongly supports that. Right. And I do that in general when I talk about evidences for God. I say that no one of these proves God. Right. But when you put these all together, you get quite a compelling case. There still has to be a step of faith. Right. But it's faith based on, I think, some very strong evidence. So in this case, at what point do you say we know enough to come to the conclusion that it is extremely, extremely unlikely that natural solutions work? For me, I'm there. I'm saying, yes, I don't know what you'll discover next week, but I think we have a lot of information right now to say that right now I think the evidence is much stronger for divine intervention than for natural processes.
Well, particularly when you couple this insight that you're bringing home, that we don't understand how chemical evolution could produce the first cells, with the fact that when you look at biochemical systems, they really do look like they have been designed by an intelligent agent, right? So you couple those two ideas together, and it really means that the most reasonable conclusion is that life was the product of a creator. That was the reasoning I had, as a graduate student almost 40 years ago, biochemical systems are these amazing systems, that to me beg the question, well, how do we explain where they come from? And when I looked at the origin of life explanations as a grad student, it's like, this doesn't work. And so the fact that they look designed and chemical evolution doesn't work pushes you in the direction that there was an agent or a mind that was responsible. And 40 years later, the case for design is stronger, and the challenges confronting chemical evolution are even more legion.
Yes, I agree. And just now, some people will say, well, you don't have to have those exact 400 proteins. Maybe there could have been something different. And I agree, but still, we're still talking a really, really low percentage that would work in an early form of life.
Yeah.
And they have to be there at the same time. That's one of my big themes when I talk about design in biology, is the idea of coordination. Even a single-celled organism has to have the cooperation of various enzymes working together, and it has to have other structures. I mean, cell membranes, we think of cell membranes as being, these kind of inert little things that exist in cells, but actually cell membranes have to have some very, very specific properties to work well. And we have that, and we have RNA and then the different molecules in the cell that the enzymes work on.
Yeah.
They all have to work together. Then when you start getting up to human beings, where you have one type of cell in the whole body that makes insulin, and all these other cells that have to have insulin receptors that use insulin, then the coordination becomes, I think, mind-boggling. And it's the reason we have so many side effects with drugs. It's so hard to just barely nudge one part of the system without messing something else up. And to me, to think, well, even pharmacologists with all their intelligence, or me trying to do some drug design with all the intelligence we try to put into it, we still have a hard time getting the desired effect without messing up the system. That, to me, it's not reasonable to say random processes could do a better job of that.
Yeah. So now, I know that you said it's really a weight of evidence that convinces you there's a creator responsible for life. But if you had to choose, and this may be an unfair question, but if you had to choose one feature of biochemistry that to you is amazing, that with that respect to that one feature, you would conclude that there is a creator, what would that be? Or maybe that's not a fair question. Maybe you would dispute the question. But
there's a lot of things I could point to, but again, coordinated processes that have to work together. It's hard enough to design or have random processes create one function.
Yeah.
But to have them work together is amazing. And I did write a paper for Reasons to Believe on collagen about how many different things had to have come about at the same time to have collagen. But even in a single cell, everything that has to work together, the balance and the cooperation, I think that is even more mind-blowing than just getting one function, which is tough enough.
Yeah. Well, what impresses me is along those lines is not just even how highly coordinated all the activity is, but even in bacteria, we're now recognizing that there has to be the appropriate distribution of those biochemical, or the biomolecules in the cell. They have to be localized in particular regions for life to exist. If you start disrupting even the internal localization, you wind up with a mess. So it's not just, functional coordination; it's spatial coordination.
Spatial. And the degree to which each one works, you have to have just enough function and not too much, or you get an imbalance.
Yeah.
And that's kind of amazing too, that you have to have enzymes work well enough to do what they're supposed to do, but not work too well that you get too much made of one molecule and not enough of another. And that also is pretty mind-boggling.
Yeah. Yeah. The degree of regulation is a marvel to behold. Well, Bob, do you have anything else you want to share with us before we bring everything to a close?
Yeah, if you want to see about some other things I've written or some talks I've given, I have started a blog, and it's called God, Evidences and Perplexities. You can put that in Google, or if you want to go directly to the site, make sure you spell perplexities correctly. You leave out the and the period. It's just GodEvidencesPerplexities.com.
Okay.
GodEvidencesPerplexities.com. And I'm tackling a variety of issues. I told you I have a diverse background, so I'm tackling everything from the types of things I talked about today to hard Bible verses, philosophical problems that people have with belief. So it's going to cover the gamut. I already have populated it with some articles and some links to talks I've given, but I'm hoping to add more shortly.
Now, are you still active speaking on university campuses?
Yes, not as much as I was when I was a professor, but I am giving a talk here in the area at Cal Poly Pomona tomorrow night. It'll be more my story. I will talk a little about evidences, but I will emphasize that my own journey to faith was based on evidences, faith, and then experiences and how the three interact. And I'll be talking about that tomorrow. And if you're not going to be at my talk, which I assume most of you will not, since this podcast will probably come out after the talk, I did a very similar talk at Ohio State, and it's there on that blog. There's a link to it.
Okay, great. Well, I really hope people check that out. It's been a pleasure having you here with us on Star Cells and God, and I'm delighted that you're here at Reasons to Believe as part of our Visiting Scholar program. So thank you so much, Bob.
Good to be here.
Yeah. All right, well, thank you for joining us on today's episode of Star Cells and God. We would really love for you to get involved in the discussion in the comments below. And remember to like the video and to subscribe for more content. New episodes of Star Cells and God release each Wednesday and are available here on YouTube and on your favorite podcast app. Be sure to share this video with a friend, and remember, the more we know about science, the more we have reasons to believe.